Basically, you have to have antigen recognition in order to have a T cell be active. So you start out with an inactive T cell, and this, for example, is an infected cell. So we know then that this is a class 1 MHC holding out an antigen that was processed, an endogenous antigen, and the inactive cytotoxic T cell, which has the CD8 cluster marker, which would be right here, will recognize this antigen-presenting cell, and it will recognize the antigen. And then you get co-stimulation, of course, from cytokines as well that helps in the activation. And this will then lead to differentiation. So when we're talking specifically about CD cluster designation marker 8 T cells, remember that those are going to only recognize MHC class 1 molecules. So the CD8 means that it only recognizes MHC class 1. And MHC class 1, remember, comes from endogenous antigens from infected cells or cancerous cells. And so these naive CD8 cells are going to become cytotoxic T cells once they bind to their antigen and have the cluster designation 8 marker recognize the MHC class 1. And that's going to lead to differentiation and proliferation, which just means division, of all of these daughter T cells. And these include the cytotoxic T cells. I'm going to talk about what exactly those cells do in a minute. Memory T cells. So you build up memory of this antigen so that next time you see it again and you get infected again with this same virus or bacteria, you already have an army built up. And then there's also something called suppressor T cells, which I won't spend a lot of time talking about. Same thing as regulatory T cells. They basically help to suppress the immune response when it's no longer necessary. I want to point out here, we're going to talk about the activation of helper T cells in a minute, which are CD4 cells. But you can see one function of helper T cells is to actually secrete cytokines. Here, the cytokine is interleukin-2, which aid in the activation of the cytotoxic T cell. Cell-mediated immunity from these CD8 cytotoxic T cells is actually very similar to the effect of the natural killer cells that we talked about with innate immunity. So essentially what happens is the cytotoxic T cell binds to the target cell, which is the infected cell, and it inserts perforin, pores, and then that is also going to lead to the insertion of granzymes, and granzymes enter the target cell via the pores, and once inside they activate enzymes that trigger apoptosis, which is essentially cell death through suicide. So in this case they're showing a cancerous cell, but it could also be an infected cell. So the function of cytotoxic T cells is to kill cells that should not be present in the body because they're cancerous, or cells that are infected with a pathogen.